So hello and welcome back. Let's get started with today's video, which is on model organisms again, the second part. So we had left off last time at Drosophila, the fruit fly, and we had looked at E. coli, yeast, and roundworm as well. And today we'll be looking at a few other organisms which are interesting. But before that, I would like to point out a mistake which was present in the last video. If you remember, I had said that the size of the genome of E. coli was a few kilobase pairs long. I think I had said 19 kilobase pairs, which is actually incorrect. Its genome is actually on the order of megabase pairs. The number which I had quoted in the last video was actually closer to the human mitochondrial genome. And we know that even though mitochondria arose from prokaryotes, uh, as per the theory of endosymbiosis, over the course of evolution, they have lost most of their genes to the eukaryotic nucleus, and their genome has become much, much smaller. So, actually, the E. coli genome is 4.6 megabase pairs long, and that of yeast is 12.5 megabase pairs long. So, that's a difference of about three times, right? And then there's the roundworm, which is eight times of that. So, we see a progressive increase in the size of the genome. But whereas Paris japonica, which is the largest genome that we know of, has 149 giga base pairs of genome, the smallest plant, the smallest genome among flowering plants belongs to Arabidopsis thaliana, which we'll also see briefly in this video. So let's move on. And this is obviously the zebrafish called Danio, and you can identify it with these horizontal striations across its body. This is actually a translucent fish. You can actually look through the organism's skin inside its body, and you can see its organs as they function. And even as the fish is growing from its egg to the adult form, you can see how the organs are actually developing throughout the course of its uh, growth as a embryo. And that makes it very interesting organism to study because you don't have to do anything invasive to look at these things. And this becomes a very good model for vertebrate development. And interestingly, even though it has just half the size of the genome as compared to humans, it does have most of the genes that we possess. And that further makes it a better organism to study as far as development is concerned, even with respect to how humans develop. So this is actually a freshwater fish. It's about an inch long. It's a vertebrate, and so it possesses similarity to us. The time it takes for the egg to become an adult is just 3 months, even though its lifespan is much longer. And each time it lays eggs, it lays around 100 to 200 eggs, which makes it easier to obtain large quantities of this organism in order to do the studies that we plan, right? And another fantastic thing about this organism is that it can regenerate vital tissues of its body. So if we can crack how it does this, how the genes are regulated in order to identify this kind of uh, trauma and then respond to it in a manner that actually completely restores these tissues rather than leading to death, that would be a very, very interesting thing for humans. And uh, the kinds of things that we are able to do with these organisms is exemplified with the example of the zebrafish because we have modified it to quite a large extent. So one example is the glowfish, which is formed by taking a zebrafish and then uh, making making a protein from the sea coral express inside the zebrafish. So here you can see red colored zebrafish because they're expressing this protein from the sea coral. And the way we are using this is to detect pollution. So when we take these organisms and leave them in fresh water, if the water is polluted, on exposure to the pollutants, there is expression of this red fluorescent protein and the organism glows red. So that is one application for it. And in fact, we can apparently buy this fish for $5 a piece. So that is something even fish enthusiasts might be interested in. But we have gone one step further, in fact, several steps further, I would say. Here, instead of coloring the whole organism with just one red fluorescent protein, what we have done is we have taken the organism's brain and we have colored every neuron of that brain. And now, during the course of development, we can actually follow the path of individual neurons as they grow, and we can see where their cell processes are ending up, what they are communicating with, and all of this in a small zebrafish embryo. So, in fact, doing this makes us able to map its lineage, right? The lineage of every cell, like in the case of the roundworm. And not just the development, but also we can use these organisms as a screening model for drugs. So, the process is simple. You just dissolve the drug in the tank in which you're growing these organisms, and then you observe its effect on them. And them being translucent, especially during the course of development, uh, things like teratogens can be studied quite well. And in a similar manner, we can actually grow human cancerous cells inside of these organisms. So if you take human cancerous cells and seed it inside an adult uh, zebrafish, it being translucent allows us to actually see all the stages that are happening when the tumor is growing. So that is another interesting thing that we can do with the zebrafish. And similarly, Xenopus is also a vertebrate and is used for similar purposes. Its genome is larger, almost double the size of the zebrafish, and closer to the size of the genome of humans. And it is present on 18 chromosomes here. And this is also a model for vertebrate development and for studying the effect of various drugs on development. And finally, we come to mice. These are the organisms that we think of when we think of model organisms and the organisms that are studied in laboratories. And mice are very good organisms because they contain so much genetic identity with human beings. So more than 99% of genes that we possess are actually present in these mice, and they actually function very similarly to what function they have in human beings. So you can potentially knock a gene out and see what effect it's having in these organisms and then extrapolate it to the humans. Moreover, there are diseases in humans which behave very similarly in rats as well, rats and mice as well, and they make really good model organisms to study not just the course of the disease but also to study the effect of potential therapies. So even though their genome is slightly smaller than us, they contain all these genes that make them really good organisms to study. Moreover, they attain sexual maturity at just 1 month of age, and their pregnancy lasts just 3 weeks. And you can have multiple generations of multiple offspring of the same pair of mice because they have a long lifespan of 1 to 3 years as well. So that makes them really, really nice organisms to study. In fact, there are mutant strains available. So, example, you want to say study cystic fibrosis, or you want to study spinal muscular atrophy. So, whatever you want to study, you might have a mouse model for it that you can buy and then do your experiments for research, right? And so far, we have seen organisms from prokaryotes to worms to vertebrates, and most of these we would consider animals, right? But we have not yet seen a model organism for large flowering plants, and that is actually a very difficult thing to find because we had seen that flowering plants have very large genomes and an insane amount of genes. Like even in this video, we had seen Paris japonica having almost 150 GB of uh, of DNA, and that makes them really difficult to study because we don't know what all these genes do. And they have several genes as well, right? So, for example, this is the simplest flowering plant that we have found, and we can use this because of the several things that will be seeing in this slide. But it having just 125 megabase pairs of genome doesn't prevent it from having 25,000 genes, and we really don't know the function of most of these. And you can imagine how many more genes Paris japonica has, right? The good thing about this plant is that it can grow in a haploid state. So, for example, if you're studying what the effect of a gene is on a plant, what function it plays, the effect can only be known in the absence of function, right? So you would like to knock out that gene, and that becomes really difficult with flowering plants because they several times have polyploid status of the genome. So knocking out just one copy of the gene does not do anything. They have 10 other copies, right? But this particular plant, if you take its pollen and grow them on a petri dish, they will grow into a plant that looks exactly like this. So they are like yeast in this manner, and you can then induce a homozygous diploidy by exposing it to colchicine. So all these are really crazy experiments which just cannot be done with human cells, or as far as I understand, the other animal models as well. So these are some great uh, things that Arabidopsis thaliana can do. Uh, Apart from it being very easy to grow, it can be grown indoors and it takes a very short while to grow. And of course, because they produce so many pollen, they are they can be grown in the thousands in a very short period of time. So all these things make Arabidopsis thaliana a good plant model for study. And suppose that you are not interested in all these organisms. You want to study a brand new organism because all these organisms are boring for you. So, how would you select an organism to study? What are the characteristics that this organism should have? So, this is a good table from Clark, and it tells us that any new organism that you are trying to see should have these characteristics. One, you should consider its location in the tree of life because we already know that we have like two, three models for vertebrate development. We have a model for invertebrate development. We have a roundworm. We have some bacteria. But the tree of life is really, really large, right? So if you are taking the efforts of developing a model organism, which would involve mapping its genome, identifying the various genes, and making a map of that genome and characterizing each gene and what it does. And this is a big effort because the effort is cumulative. So you do some work and maybe some other researchers do some work, and all of this work is now available for a third researcher to actually work on this. So you would want to actually take an organism which has not been represented by the commonly used model organisms available today, so that we can cover a wider array of the samples from the tree of life. And when we are doing this, obviously, we should be able to clone the organism's DNA. We should be able to identify the genes and understand what each gene does by observing the organism's development, being able to knock a few genes out and then seeing what effect it has and such. And we should be obviously able to grow them in large numbers, and they should ideally have a short life cycle so that we can observe the entire uh, life cycle and the effect that the gene plays at different stages of development. And of course, we should be able to either overexpress or knock out genes and see the effect that this has on the organism. And if we have close relatives of this organism available, then we can see how the genome of this particular organism, the model organism that we are trying to make, differs from its close relatives and how that translates to the phenotypic difference. So that would give us some clues as to the function of the different parts of its genome. And once we are doing all this work, we make a genetic map, we locate the genes, the various genes on each of its chromosomes, and then we make all this work available for everybody so that anybody can work on this organism and expand the collective knowledge that we possess. So I think that would end the video that I wanted to make today. And I wanted to take some time to actually discuss the philosophy that I'm trying to implement here. So I am a medical geneticist, and this would be the book I would, I think, try to start with. This is a book which a lot of people read for medical genetics, Thompson. But I felt that it would be really cool if we could go through the cell biology and basic genetics and molecular biology from Clark and cell biology from Dalton before we move on to medical genetics so that we understand these concepts better. Because most of the time in medical genetics, the actual genetics part gets downplayed because we have so many diseases and so many patients to care for. But all the same, the basic genetics remains my favorite, and I would first like to explore that, and then we will eventually make videos on the various diseases and medical genetics aspects as well. So I think that is all. Thank you.